So yesterday also the hand DP measurements which you used is basically the fluorescence emitted by algae or whatever the chlorophyll molecules, whether it is plants or algae. You use the fluorescent signals from them to study how the photosynthesis is going on. So here also same in PAM also we use the fluorescent signal only. The signals which emitted from the photosystems we use to study how efficiently it is working. This is the basics of all this kind of biophysical techniques we use, which is differed only whether it is we are using the fluorescent signal or the absorbance. That's it. So PAM basically we use PS2 fluorescence to study, but for PS1 we use the absorbance. Why? Because for PS1 in room temperature PS1 fluorescence is difficult to detect. It's negligible. Like in PS2 fluorescence, it's the largest. So the one which is from PS1 will get overlapped and you won't be able to detect PS1 fluorescence when there is PS2 fluorescence. So in room temperature it is not possible, but we will learn another technique called 77K fluorescence, which is like low temperature fluorescence. You use liquid nitrogen temperature, there only you can detect PS1 fluorescence. So here you will be learning PS2 fluorescence to study the photosynthetic efficiency. I'll show few slides. So what is chlorophyll fluorescence? Chlorophyll fluorescence is not only red and beautiful. Chlorophyll fluorescence is the fluorescing light. The emitting light is actually red, okay, detected, and which is not red and beautiful, which is also can be used. The large signal which gives properly measured and analyzed can give detailed information what is going on inside the photosystem. Okay, then so one more. So you see, when the light harvesting complex or the photosystem absorbs the light energy, it converted into three, or it can go to as fluorescence, it can be done photochemistry, and the remaining can go as heat. So this is the three, and the sum should be 1. So whatever the portion you get as fluorescence, this fluorescent signal can be used for studying the other part, photochemistry, because the sum is 1. So whatever the light is absorbed, which will be either fluorescence or it should be photochemistry, the part which we are learning, the electron transport and final ATP and NADPH. Otherwise, if it's excess, it will be emitted as heat. This is the 3 components. So we are going to use the 1 component, which is fluorescence, to study the other components. Now coming to dual PAM. So which is pulse amplitude modulation. Why it is called pulse amplitude modulation fluorometry? So basically there are 3 light components in dual PAM. One is measuring light, which is basically the modulated light, or you can say it's a very weak light. You can say like 1 micromole Einstein, very weak, but this will be continuously probed. Understand, there will be a probe to emit the light. So this emitted light will be pulse amplitude modulated. Why it is called pulse amplitude modulated? This will be continuously emitted like 1000 pulses per second, you can say. So which is like a frequency it will go. That's why it is called pulse amplitude modulated. And this high frequency, which is like per fraction of seconds, thousands of pulses of small weak light will be continuously emitted. For what? This light can excite the chlorophyll molecules, which can only get fluorescence. They can't do photochemistry, which is very weak. So this light cannot be used for a photochemistry or electron transport or anything, which can only excite just fluorescence. So this will be the probe, or you can say for every experiment we will have a baseline. So this will work as a baseline for the system itself. So this will be function as a reference, you can say. So this is called measuring light. So that's how PAM detects the signal which is coming from the plants or whatever the system we are working with. That's why it is called pulse amplitude modulation fluorometry. And by shining a beam of light on a leaf and measuring the light re-emitted by the leaf, it is possible to measure not only the photosynthetic activity, but also several underlying regulatory mechanisms that control the photosynthetic processes. So doing a lot of mathematical expressions within the system using the single large signal from the fluorescence, it can tell you a lot of parameters. Not only yesterday you must have studied only Fe by Fm, F naught and whatever the OGIP, but here it will tell kind of quenching, non-photochemical quenching, different kinds of quenching, zeaxanthin based quenching. You can say the acceptor side limitation of PS1, PS2, what is happening its acceptors, what is happening it's in the donor side. These all kind of parameters you will be getting from a single measurement. So I told there is 3 components. I only explained measuring light because which is the basic for dual PAM. That's why it is termed as PAM. So now coming to the second. So I hope you understood what is the measuring light component. So whatever experiment you are going to do in PAM, measuring light will be on. This frequent signal can only be detected by the PAM detector part. So next one is, so we call measuring light as the probe, and the second one is actinic light, which is like the worker. I told you measuring light can only give you the fluorescence from only the chlorophyll fluorescence. They can't make the chlorophyll molecules to work for photochemistry or to the electron transport or anything. They can do only a very weak light. But in case of actinic light, we can call them as the worker, and they drive the photosynthesis. When you give an actinic light which is very intense, you can give different intensity of actinic light starting from 12. Right now you will see down how it is working. We are giving from like 12 microeinstein, you can go up to 1000, or if you want to go more, you can go more, with an interval of time, like 20 seconds, 30 seconds. These all you can program. So giving this interval 20 seconds or 30 seconds, you increase the actinic light, they will start photochemistry. So measuring light is already giving a pulse. Now you will give intense highlight, not highlight, like increased intensity of light, which is called actinic light, and they start photochemistry. This opens and closes the PS2 reaction centers by exciting the electrons. And also before giving the actinic light experiment, you give a dark adaptation also, so that all the PS2 reaction centers will be closed and directly you are going to saturate them with slowly increasing the light intensity. Now coming to the next light component is the saturating pulse. The name itself is saturating pulse. What does that mean? It can completely saturate at a time. It will be very high intensity light. Normally we give here 3000. You can go up to, like, I don't remember the exact range, but usually it will be more than 8000 micromole photons. And this is a momentarily close of PS2 because you are giving a very high light intensity. So for a momentary, the PS2 reaction centers completely get close, and you get like temporarily stop the photochemistry because you are giving a very high intensity of light. Everything is saturated now. You are giving that much high intensity everywhere. Everything is saturated. That's why it is called saturating pulse. And this stops the photochemistry, and at that pulse, which you just imagine all the light harvesting complexes are ready, you're giving a very high light intensity, they just absorb the light and the electron transport happens. Everything is saturated. So you just get one peak and then everything is closed. So this is the maximum fluorescence PS2 can give you. That's why you use a saturating pulse. Okay. So depends on experiments, you will use these lights according to your need. You want to do it like the complete PS2 reaction center. You just want to see the maximum fluorescence, or sometimes you do some kinetics also. Like not only this, you can do like what is basically kinetics means, how the electron transport is going. If you again give a saturating pulse, what will happen? These kind of experiment also you can program according to your purpose. So there are a lot of options within dual PAM you can use. Today we will only cover basic light curve, which will give you a lot of parameters. You will see that. And you see, I was talking about the saturation pulse. The graph itself says F naught will be, you are now familiar, I guess. So it is the initial fluorescence. In a measuring light, F naught signal will be in that. But you are suddenly giving a saturation pulse, you get the maximum peak, and that will be the maximum fluorescence you get from PS2. So these light components are clear, or you have any doubt, you can ask. Anything needs to be repeated? Okay. So now what is the, we are not going to use dual PAM, all the parameters and everything. You can use different probe. You can study individually cytochrome B6F. You can study the kinetics of PS2. You can study kinetics of PS1. Then you can study ATPs in this activity. These all can be done using dual PAM. If you have the proper probe. Why? For cytochrome B6F, you may need a particular wavelength. Everything gets excited in a particular wavelength. If you have the probe, you can do all this experiment. So basically we work with P700 probe, which is for PS1 kinetics and all we do. Then normal PS2 fluorescence we use. And also we have P515, which is like to study electrochromic shift. Basically carotenoid spectrum changes we study, which is bit deep we do. But today we will be discussing about light curve. So I will be only discussing light curve, what you are going to study. So in a light curve, or it is called a rapid light curve, in dual PAM measures how photosynthetic electron transport and fluorescence parameters change as actinic light intensity stepwise if you increase. I told you 12, you can increase to 20, 30, 50, whatever, and you can go up to whatever you need. And which always start with a dark adapted leaves, as you did with handy P, you keep for a dark adaptation. And usually why you are doing a dark adaptation, because you will get the F0 also, and just giving a saturation, you get the maximum fluorescence also. And the instrument increases, already I told, with a period of short 30 seconds, 20 seconds as per your program. And these will be predefined steps within the system, and you can give the actinic light increase from 0 to 1000 micromole. In some papers, you will see more than also. According to your organism, you can change this programs as well, either 1000 or you want to go up to 2000, whatever you can do. And during the entire sequence, the measuring light stays on to record the steady state fluorescence, because the dual PAM instrument always measure the measuring light signals, not from the actinic light, not from the saturating. Why? Because you are giving like a frequency of like 1000 pulses of very weak light in a fraction, which is like a frequency. And this frequency will be fixed in dual PAM. In the, we showed you the part. If you see the part here, this one, dual PAM. So this is the signal detection part will be happening. So if you see here, this is the detector part, and this here you can change. You can change both this part. So here now it is fixed at the P700 and NIR. When you want to study the PS1, so you keep this probe for emitting the light, and you will keep the sample here in between. So the light passes, and which get detected here. So this detector listens to whatever signals it come, whether it is measuring light frequency or it is actinic light, which is very high. So, but now when it comes to this part, there is a signal processor. This signal processor will only take whatever the modulated light signal is coming from here. But even though the modulated measuring light, which is the modulated light, is coming, when there is a, okay, we are giving an actinic light at one point. You keep it 30 seconds, you are giving an actinic light, but background measuring light is going on. The frequency is 20 kilohertz if you keep. Okay, so it's going on, and you are giving a light on 30 seconds. This time, this frequency will change. Whatever the frequency it was going on, it's a continuous. But suddenly, there is an actinic light fluorescence is coming, there will be a change in this frequency. So again, this processor is going to process only that change in the frequency in measuring light, not the actinic light. So it should be always on the measuring light. They never process actinic light or background the fluorescence from actinic light or the background actinic light or saturating, nothing. They always listen to the measuring light. When there is this part, always there is a fixed frequency of measuring light. The change in this frequency will be detected and amplified as the fluorescent signal. So if measuring, so you come here, F0 measuring light is going on. Whatever the graph you got is very high peak, which is the change they got in the measuring light actually. Measuring light fluorescence frequency change, not the one. Yeah, that is why it is called PAM, pulse amplitude modulated. They never detect the other. So in light curve, so that's about light curve. Now you are going to do light curve with increasing light intensity in a time period of illumination, and you are going to give actinic light in between like 30 seconds or whatever the predefined time, and you are going to get electron transport rate, what is the PS2 yield, what about the PQ pool, what is happening with the BQ pool. These all will be, the signal will be amplified and you will be getting these all results from a single signal. So we will see now how it is, and finally you can plot the graph like you can see here, electron transport rate and non-photochemical quenching. I think more than 10 parameters will be studied using only light curve signals, and you will see down that one now. That's it. And if you have any doubt related to the theory part, you can ask now, or else we can go down. T, after T. Principle is basically fluorescence. The fluorescent signal they can detect, but here they can process it in a more advanced way so that we are getting more accurate and more parameters here. You get the same F0, FM, everything here also. As I said, even in this frequency, very minute changes also in this frequency. Yes, but in HandyP you can't. In HandyP you can't do PS1. Yes. Okay. PS2 fluorescence can be detected, but in case of PS1, if you want to do in room temperature the changes, PAM detects the absorbance, not the fluorescence. So the emitter will be giving like 830 nanometer or something. We have a probe very specific. In 830 far red, only PS1 will get excited, not PS2. This is principle we know. So we give this light and the PS1 only will absorb this. Once PS1 absorb this particular wavelength, there will be absorbance change of PS1. Once it get excited PS1 and non-excited PS1 absorbance will be shifted. This will be studied by the same signal processor and you get the data. So you should always remember in PAM, PS2 fluorescence will be measured, PS1 absorbance shift will be measured. You need this PPT, or you can just type dual PAM, or else I will share. If you need this, I can share, or else there is handbook, all the details will be there, what are measuring light, what are components, everything. We can share that better than this. Okay, sure. Even if it is very small thing also, you can ask, because first if you don't understand PAM, very basic thing, then you won't understand what we are going to do further and all. So even if it is very small thing, you can ask. Oh, this graph. I think explaining this graph will be better. We will do no after doing the same we will take here. Then we will plot, then you will understand more better. This is basically you see light intensity is increasing 0 to 2000. On a time basis you will increase. And what is happening to the electron transport from PS2, how it is going, photochemistry, what is happening. And down is non-photochemical quenching. With the same fluorescent signal, we will be able to see the quenching also.
On what conditions you are growing, what stress you are doing. Sometimes there may not be change in donor side and acceptor side limitation. That means the plant's electron transport is in complete harmony. The amount of electrons that is passed on to PS1 to PS2, and that much electron, it's again going to the downstream process. So it's not giving too much. But just know the basic structure will be this way. Right? So YND and YNA is dependent on your PS1 as well as Y1. Y1 means the yield of the PS1. So yield, you see, this is the yield. Just consider, don't consider this point. Start from here. So if you see the yield, it initially is from this region and it is dropping. Why the yield drops?
The limitation increases.
No. Why this? What are these points exactly? If you want to go. So basically it is running around 1400 microingstein, right? The last actinic light was around 1400, this point. You see, x-axis is PAR. PAR means photosynthetically active radiation, microingstein. So around 1400 or 1401 or 2 might be there some point. So this is your last actinic light intensity. This will be your lowest light intensity. So under saturating pulses and if you increment the light intensity to a higher point, then the plant's yield will drop. Any plant's yield will drop because 30 seconds you are giving time. In that 30 seconds with a saturating pulse, every saturating pulse, you are closing all the reaction centers. Then again in the actinic light intensity, in the lower actinic light intensity, some of the photosystems are again they are opening. Opening means again there is like oxidation is there. So if a higher actinic light is given along with the saturating pulse, the amount of reaction centers that are going to open may be less. You got it? So under lower conditions, lower intensity light conditions, the actinic, because suppose you start from maybe 30 light PAR, okay, 30 microingstein you started. Now comparing with 30 to 1400, under 1400, it's like how much times? How much? 3 times? 3 or 4, whatever it is. So 1400, 3-4 times it is higher. So under 30 microingstein and under 1400, and also the plant's adaptation is only 30 seconds. We are not giving enough time for the plants to adapt to that particular light intensity. Only 30 seconds. You can also go lower. 30 seconds, 10 seconds you can go, depend, but average protocol is 30 seconds. So after 30 seconds, again a saturating pulse to record a point, then a higher light intensity for 30 seconds. So increasingly you are incrementing the PAR intensities, and with that PAR intensities, your reduced reaction centers quantity is increasing. Got it? And if it is increasing, the performance will be low or high? It will be lower, because I already told you, if a photosystem's reaction center is in already reduced state, can it accept any more electrons? It cannot. So when will it accept electrons? When it gets oxidized. So if it is in reduced itself, so it cannot operate anything, right? It cannot accept the electrons. And if it is in reduced state, that means the electrons are not exiting that reaction center. So if it is exiting, then only it will get oxidized. So with the higher PAR intensities, so the amount of reaction centers that are supposed to be open is, the quantity of that is getting less by less, means decreasing with every higher light intensities. Okay, that is the reason this means this point are dropping, right? So in every type of plant, in every stress, this drop will be there, but in under stress the drop might be more prominent. You understood? So in case if I take a stress plant, this drop might be even more. Initial drop will be there. Okay. The later drop may not be that much, but the initial drop will be quick. Got it? Under stress, if you'll compare these two things. So Y1 and Y1, YND, YNA, you understood? Then PS2 associated parameter is, I think, Okay, these, and last one is your ETR1. Okay, this is your ETR1. I'm just showing what is it. So this is your ETR1. So ETR1 does not, in light curve, ETR1 does not mean your electron transport of PS1 itself. It represents the cyclic electron transport as well as non-cyclic electron transport, linear as well as cyclic, right? Because through PS1 both the electrons will pass. One is from PS1, PS2 to PS1, and other one will be through cyclic. So PS1, this light curve will represent your both electrons. Don't confuse that this represents only the cyclic electron transport. This just represents the amount of electrons that is passing through it. So it can be from PS2 as well as PS1. So anything can be contributing to it. So that is the reason the ETR1 is always higher than ETR2. See, if I go to ETR2, this thing you see. So why ETR2? Because ETR2 is only passing your linear electron transport electrons. Initial charge separation will be there. After the charge separation, PQA, PQB, then cytochrome B6F plus and then to PS1. Got it? But PS1 will be coming first. PS1 will be getting electrons from PS2, the same cytochrome B6F pathway, and other one will be through cyclic electron transport. Again, it will be returning back. So it's getting two kinds of electrons. One is through linear, one is through cyclic. So both of these are combined. That is the reason always the ETR1 is higher than ETR2. It should be that way. Okay, ETR1 should always be higher than your ETR2. So don't confuse that. ETR means it's representing your cyclic electron transport. It represents your how much electrons it's passing through the ETR1. Got it? It does not differentiate between cyclic or non-cyclic. So this one is your ETR1. Got it? Also these four parameters like Y1, YND, YNA and ETR1. All these four parameters are you related to PS1. So rest other parameters, the basic parameters I am explaining first. Other parameters, one is your ETR2. ETR2 I already explained, that is just a linear electron transport. If the charge separation is proper, if everything is, the plant is not under stress, ETR2 will be higher. Usually this condition should happen. So if your ABOFM is low, okay, so usually it should show change in yield and ETR. Got it? So this is your ETR2. And this one is your Y2. What is that? Y2. So this is your yield of the Y2, same like the one I told you, the same way it is plotted. Y2, Y1 the same way it's plotted, ETR2, ETR1 the same way it's plotted. Okay, only the ETR1 will be higher than ETR2. So this is Y2. Then comes your YNPQ. Now already that day I told you heat can be dissipated in the form of YNPQ, but there is another way of heat dissipation, that is YNO. Okay, so YNPQ is a form of regulated form of heat dissipation. For example, if a plant is performing properly, you are then suddenly giving some kind of stress. Now if the plant is kind of adapting to that kind of stress, it will try to dissipate that energy in the form of YNPQ. Now if the stress is too much for the plant and it can't handle that much amount of stress through its YNPQ, that amount of energy will be taken over by your YNO. YNO is a kind of non-regulated form of energy dissipation. Got it? This is non-regulated way. You cannot regulate it. If the plant is not able to regulate any kind of stress, that stress energy will be taken over by your YNO. So just by looking at your YNPQ, you cannot for sure say that the plant is performing good or bad. You have to check with your yield. Yield means Y2, right? Y2, YNPQ and YNO. Got it? Y2 is your yield. If the yield is dropping, then there is some change, and if YNO is increasing, usually YNPQ and YNO are inversely proportional. If YNPQ increases, YNO drops. If YNO increases, YNPQ drops. If a plant is having a higher YNO and it is having a lower YNPQ and the yield is low, so is the plant under stress or not?
High.
I am talking about higher YNO. Non-regulated energy dissipation is high, yield is low, and third one is your YNPQ. YNPQ is low. Okay. So what condition, what can you infer from this?
Why NO is better than plant is under high?
Yes, under stress. So now because I included Y2, you may infer that it is under stress. Now if I remove Y2, if I just say if your YNO is higher than your control and YNPQ is lower than your control plants. Control plants means non-stressed plants. Okay, so in your non-stressed plants, the control is in your non-stressed plants, the NPQ is higher. Under stress plants, NPQ is lower. And under non-stress plants, I'm talking about stress, non-stress, but if I just give you two plants, one is having higher YNPQ, one is having lower YNPQ, one is having lower YNO, and other one is having higher YNO. That means which one is under stress? Higher YNO. So always remember, YNO is higher means it should be under stress. If YNO is not higher, YNPQ should be higher. YNPQ is higher means the plant is efficiently dissipating the energy. Got it? So under too much stress, that YNPQ energy, if it is not able to dissipate, if the YNPQ apparatus itself is already damaged, that energy has to be taken over by YNO. So YNO will increase. That will take over the energy from YNPQ. That will increase. Okay, and because of that energy taken over by YNO, YNPQ now will drop. Got it? So just by looking at YNO, YNPQ, you cannot tell that it is under stress or not. You have to check with Y2, YNPQ and YNO. Got it? So usually these three things are combined, and if we'll go to any good research journals, usually the YNPQ plus YNO data plus Y2 data equals to 100% or unity. Okay, here it's 100% because we take it 100%. So if you calculate each points, each points YNPQ, you take this point, some reading will come. This is your not it here. Okay, so these points you take all these readings. Some reading will come here, some reading will come here, and some reading will come here. Just you add up all these three, so you'll be getting around 100%. In some PAM machines, they go for unity. Instead of 100%, that will be 1. Got it? Similarly, these will be in point. So this is basically y-axis is in percentage. Got it? That's why all these three will be unity. Similarly, y1, ynd, yna will be 100% or unity. Got it? So these three things you already know. Then comes your YNPQ by 4 is a like normalized form of NPQ. So NPQ has many components. I am not going into details of everything. So it's a kind of normalized form. Usually we don't plot NPQ by 4. So we'll avoid that. Then ETR2, ETR1. Then coming to the quenching parameters, QP. So QP is your photochemical quenching. Coefficient, Q means coefficient of photochemical quenching. This is also directly related to your however the plant is performing. If the photochemical quenching is good, that means electron transfer is good, the QP will be good. So if I plot QP here, similarly QP, QN and QL, these are all associated with your PS2 or PS1? PS2. Okay, so PS2, what parameters are there? One is Y2, second one is YNO, third one is YNPQ, then comes your QP, QL, QN and NPQ by 4 and ETR2. NPQ by 4 you usually don't plot. So if you go to keep QP, this is your photochemical quenching, coefficient photochemical quenching. It will be, the plotting will be just like your yield. But usually this is used in a supplementary data. If you are giving the yield, just to make sure that you are giving it right, these graphs are plotted in your supplementary data. So QP, let's see, there are two types of photochemical quenching. One is depending on the scientist who did, one is lake model, one is pool model. Got it? QP is through pool model, QL is through lake model. So if the photosynthesis systems like LHCs are properly interconnected normally for higher plant systems, usually higher plant systems, they take QP, pool system, right? And for algal systems, some like marine alga or depends. Some people also take QP for plants, the algal systems also. QL is also kind of photochemical quenching, but that is through lake model. The calculation is different. Lake model has a different calculation, then pool model has a different calculation. You don't need to stress over that this software will directly calculate and plot. So if you are taking, you can either take QP or QL. No need to take both. Got it? So QP and QL, you see both are almost same, but depending on the calculation type, the numbers are changing. Then comes your QN. QN is your what? Coefficient of non-photochemical quenching. Okay, so there is different types of quenching. First, maybe I'll talk later. Just for example, quenching can be many types. First, at the last will be zeaxanthin dependent, QZ, QI is inhibitory quenching. I'm not going into details, otherwise we'll just, so just remember whatever you need to plot.
Maybe you will show how to plot, right? Maybe that time you can give more explanation.
Okay.
Maybe they can do the maybe hands-on.
Yeah, so this thing, once you do this, run it. After the running is done, just make sure you click everything what you want to export. So I'm just clicking everything to export. Okay, so this fitting parameters you don't need, last two. So all these parameters apart from the last two, everything is, then you just export. So export will be in CSV format. So Excel file it will be exported. Let's go to desktop. Okay, now it is saved. Now if you want to go for a second leaf, just go to here, file, go to new report, go to yes. Got it? So it will override whatever the readings were taken. So anybody wants to do?
Can you explain the algal system?
Yeah, algal system, the basically everything is same for algal systems. The only difference is that because these are cultures, in liquid cultures, you have to use a cuvette. Okay, so minimum 1 ml is fine, enough you can take. These two systems will be fitted over these two regions. That's it. Nothing else. Nothing will differ. Only the algal culture you need to put. Just because it's algal culture to dark incubate, you can do directly. Place on it, and this two will be fitted on this one, tighten it, and the running procedure will be exactly same. Got it? In algal culture, you may not get P700 because if some cultures are growing in salt media and all, there will be some signal changes. So, but PS2 will record very properly. Got it? So, anyone wants to do light curve? So don't expect the Fe by Fm to be more than 0.79 or 8 because it's already exposed to light. So just for demo purpose, I'm showing you. Yeah. Great. Okay, so this is the first, I'm closing it. Okay, just open this program, P700. This one, dual pan. P700 and Fluo. So this. Okay. Yeah, just maximize it. You can sit. So it depends on what you want to do. Since it is leaves, you can go for Fluo and P700. I already selected it. So detector, you just see DR, this is not connected. Detector 2, you see nothing is connected here, so it is not connected, NC, and rest of the things is fine. Then you have to go for measuring light. Okay, balancing you have to do. Then go for this one. Okay, now balancing is done. As long once you pressed it, this measuring light will be on. Now you go to light curve here. Okay, so just click start. So why I didn't tell to do the zero offset? Because it was starting from 0. Okay, that's why I didn't do the zero offset. If it is plus 0.05 or minus 0.05, that is acceptable. If it is more than 0.10, yeah, that's, you have to do that.
So the interval program you have already...
Yeah, no, it is pre-fed. Normally the light curve will be pre-fed. You can directly place the leaf and run the program.
So if I want some other intervals, can I change?
Yeah, you can change it. You can change. For that, writing a script file is necessary. If I start to explain that, it will be very long. So for now, this is by default, it will be there. 30-second exposure will be there. After 30 seconds, saturating pulse, then a higher incremental power. So now it is running in 48. So you see every time it will flash a light. It's recording now. You can switch off one light, this big one. Yeah. So this now the actinic light running is this one 48.
Okay.
So you see a red light will be flash. So that will be your saturating pulse. So now the power increased. So the increased power intensity is displayed here 61.
What is this? Plus 0.384?
Oh, that is the voltage change, nothing else. Because you have to, I told na, you are getting it to a baseline balance thing. While running a light curve, this should be more or less stable. It should not deflect too much. If it deflects too much, then there is some problem with this machine. So now the power is 111, right? So each saturating pulse will record a point.
How long do you take the reading for one sample?
It depends. If 30 seconds duration is there, then like 10 or 14 pulses you are giving. Just multiply with 30 seconds. That much seconds you will be having for each sample.
Okay. And I have one more doubt. So we are taking FO in the start. After that, the probe or the measuring light is already on. Right? So we have little bit of light. So the FM would be affected because
No. Measuring light does not affect your FM. Your saturating pulse will record your FM.
No, because earlier we are keeping it in dark.
Yes.
And then since we are giving low light.
No, measuring light, I told na, measuring light does not induce any kind of electrons. It's a very low light in the 5 microangstrom, within 5 to 10. 5 to 10 means maybe under this you will be getting 1 or 2. So 5 is very less. Got it? So you can click everything if you want to check. You can click one by one.
Sir, so if I have to read it like, so at 50 power units of light, so it's the yield is 20%. So how do I?
Usually light curve is taken as a whole, not a point.
Okay.
Got it? It has to be plotted in a range from 0 power to 1400 power. So it will be in a curve. That's why the name is then light curve. So how the plants are performing to each incremental light steps. Obviously, better performing plants will have a better one, and the plant which is not performing well, the decrease will be very quick. Exponential decrease will be there.
So here we are keeping one sample only, but yesterday handy P. There is one only test come, but here many.
Yeah, this you can keep one leaf at a time, but this one is more precise. It's far costlier than that one also. So this you can measure a lot of parameters. Light curve is just maybe 5-10% part of PAM. So you can measure everything, cycle electron transport and everything in it.
Yeah.
You have to make a script file for that. But by default, the light curve is the first way to
I'm
go.
very confused. Actually, what exactly we need actually for
Exactly means like
exactly means okay, but our experimental purpose. If suppose you want to see the for PS1, PS2.
yeah, see PS2, PS1 basic things you can get from HandyP. FBF not, that is the primary step. If you want to check whether a plant is under stress or not, that is the primary step. You just check ABFM, that's it. You get to know that it is having stress. Now if you want to further analyze it, whether the CET is getting upregulated, how the electron transport is shifting, whether the electrons is bottlenecked from PS2 to PS1 or after PS1, that thing you can do it from light curve.
Okay, but whether it is giving the actual results, the interpretation, actually any calibration is required or
No. It is already actually recently we did the January, February, we send it to Germany. Calibration is not done here. We have to send it to Germany. Yes, that they send it back. So it took 2-3 months. So it is already calibrated.
okay.
And before running, this balancing is the calibration of
Is there any
P700.
control is there for running? But is suppose actually some control, some calibration and all. But whatever they said, we have to listen, or we have to. Is there any control? Control.
No, calibration means for what you need.
No, actually, whatever you are saying, actually some experimental. That experimental part, actually,
Yeah,
the live experiment.
means like you are telling if you want to change some parameters or something.
Yeah, change parameters and suppose some you are telling electron transporting and these and all those things. No, actually, that how to.
So no, once this is run,
Okay.
I will explain it up, how to export it and how to plot it, everything. So you run 2-3 samples, you just take the average of these 3. This will be just numbers, nothing else.
Okay.
In x-axis the power intensity will be there, y-axis the numbers will be there in percentage. Y1 there will be some numbers. You just plot it in Excel sheet. You can just plot any other leaf under stress. Just compare these 2 graphs.
Okay.
You can get this normal comparison, not a big deal.
Jyoti, can we take the technical replicates once we are?
You can take. These are all technical replicates only. The one I'm taking, this is all technical replicates.
I mean 3 or 4, how many?
No, at a time you cannot take. One by one you have to do.
So that's why I'm asking. We are take from the dark actually. That many plants are there. So can we take the after one plant?
Yeah, you can take. See, I told the minimum time of dark adaptation is 25 minutes. So if you extend the 1 hour also, it's not fine. It's fine. At least for plants, it's fine. You can extend to 2 hours also. For slow kinetics, we dark incubate for 3 hours. So it's not a big deal. But minimum amount has to be qualified. If it is 25 minutes, 5 to 10 minutes, 20 minutes, 30 minutes of 1 hour gap also, it's fine. Just we are making sure that PS2 reaction centers are completely open. After 20 minutes, the reaction centers are all oxidized state.
So in that pot actually, many plants are there. So if you want to take 5 plants are there. For example. If you want to take the each and every plant. So now we can able to do that, na? Or again we have to kept in dark.
No. See, what I do.
Clips like handy.
No, clips are not here. So what I do is I grow single plant in one pot.
Okay.
So I dark adapt all the plants at a time. So for light curve, I'll take that single plant, take a leaf of it, take the readings. Then after the readings is over, I'll take the second pot. That will be having single plant. Then take that way, I'll be having very less margin of difference. Okay, that will be technical replicates, but I will get margin of difference. I'll be getting very less that way.
What about the biological replicates?
Biological replicates you want to do, you can do. You can just repeat with after some days. If you are having the same plant, you can repeat it. Then you can take average.
The suppliers actually for this.
This supplier is actually the company is from Germany only, but the supplier is Indian. So supplier means they will procure from them, and like the costing maybe 50-60 lakhs.
50-60 lakhs.
Yes.
In our lab also there. My lab also there.
Yeah. No, another one version is there. Dual PAM, I think a higher version. Something. That one is like maybe 90 something.
90 lakhs.
Yes.
90.
Yeah.
Reaction. So you need to read it how in stress conditions
Could you be talking about it?
sometimes stress to stress
SMT
OJIP, SMT. SMT, it's better that in dual, this thing you can get, HandyP. But for that, you have to go for 5 minutes light intensity. The initial we did was for OJIP, that was 1 second. Okay. If you want for SMT curve, the same program has to be run for 5 minutes. So for 5 minutes, OJIP will be there. No, you can't do it here. This is basically a more detailed version. SMT can be done in HandyP. You just have to extend the time frame. Initially it was done for 1 second. Now it will be extended to 5 minutes. So you can get for slow fluorescence and plateau phase also you can get it. Okay.
Quenching has several components.
Okay.
You can get
It's
QI,
done.
QP.
Okay. Once it is done,
I'm not taking photograph.
okay. Fine.
Yeah, many outcomes.
If you want
Yeah, it like all like 13-14 parameters are there, but you don't need all parameters. You can take 9-10 parameters. You can take proper.
Amma, it's saying there, one would have
Sir, what are these?
These two are actually fitting parameters. Normally, if it are some changes there, na, it's not very smooth. So there are different models. One is Platetol and one is EP. You don't need fitting parameters. You can directly plot it.
What is this, sir?
These are like P700 states. Okay. PM dash, oxidation rate. These things you don't need. Normally, it will just display the numbers and these numbers will change according to each point. So this is for the last point. You just go to export.
we'll take there, then Excel you can just, it will come in text format. You need
Okay. Okay, it is saved. So anyone else wants to do?
QP and QN.
QP?
QN.
QP is here. QP is your pool model. Both are quenching models, photochemical quenching. One is pool model, one is lake model. You can take either of them. QN is a non-photochemical quenching coefficient. It's similar to NPQ only.
It means all these stuff are
Yeah, QN, there is many ways to dissipate NPQ, right? I told zeaxanthin dependent, inhibitory.
just calculating on the
HandyP is very initial. So HandyP is just frozen, nothing
There
else.
complete transfer of letter
Yeah,
was taking place.
yeah.
But there was no transfer.
There will check the ABY FM, that's it. Only this point it will check. ABY FM, FM, F0. That's it. Nothing else it will check.
Okay.
Okay. So anyone wants to do? Okay.
These techniques, if you want to understand and interpret data, you need to read a lot about quenching part, different components of quenching. There are 5 or 6 components of quenching.
Okay.
సెకండ్స్ కి పెట్టుకోవచ్చు టైమర్ సెట్ చేసుకోవచ్చు అలా ఫ్లాష్ అయినప్పుడల్లా ఒక రీడింగ్ వస్తుంది ఆ రీడింగ్ మనం ఆటోమేటిక్గా సెల్ఫిటేషన్ కూడా వచ్చేస్తుంది ఫస్ట్ మనకి ఫ్లాష్ అవ్వగానే ఒక
Like yesterday only, that leaf part only, sir.
Yeah. Whole leaf you can get. So just it doesn't, dorsal part has to be this way, emitter. Dorsal part means you tell it abaxial, adaxial. Adaxial side has to be towards emitter, right? Yeah, you just open the software.
Same plot there or
Same plot only, by 2.
Here you don't put the picture in groups, or you go up.
No, upstairs it's better that you just plot it. Okay. Okay.
Sir, tomorrow you planning Irga from
Irga,
SHT, SKG, Skill
Which one?
Development Center.
I don't know. I have to talk to sir regarding that. It's already selected, right? So DR is already selected. So you just go to measuring light, then balance it here. Okay. Then it's on. Go to light curve. Here, light curve. Click.
Zero there, somewhere.
That's fine. 0. It's on range. It's in fine range. Go to light curve. Then click start. Okay, done. It's starting already. So the first flash will record MVFM. Okay. Because no light is already, it is dark adapted. It's same like HandyP, first flash. After that it will take the light curve. Done. Last maybe. 3 to 5 replicates is more than enough for your technical replicates. No, while exporting you can just, if you'll export also, this too will not come anything.
Those are like standard.
No, these are fitting parameters just to check how the fit is proper or not. Just to some people they fit the ETR. ETR will be some zagged kind of thing. That time they fit, but it is no need of fitting.
Done. They're just
So instead of planning. Research background.
So is there any difference between Y, NPQ and NPQ? Just sometimes I feel
No, NPQ and YNPQ, that is just an interchangeable term. That's it. Yield of NPQ is YNPQ, NPQ is just non-photochemical quenching. Usually we measure the yield only. Okay. Just like the yield of PS2 represents the PS2 action activity, that's the same way YNPQ and NPQ. Swell, water bottle is there. I'm thirsty.
don't plot all parameters.
Oh, done.
Plot major parameters and show. Anyway, this much they won't understand.
Light curve only, just NPQ and all.
You can't control that.
Before this
Manual method. In analysis culture, when you work with this voltage fluctuation will be too much because they are always moving and there will be hindrance in the passage. So this voltage fluctuation is too much, and you will get these curves are so smooth and good, right? But when you work with the liquid culture, you won't get like this. I will go.
You want to go?
Yes, I want to.
It's 5. If sir would have been here, he would have said till 6. Sir will be there in lab or not?
I don't know.
He will come back. He will drop Yuchiro and come back again.
I want to go.
Where?
Secrets.
Kantara.
Kantara is there. I'll go change it.
I'm not going to give theory of Irga. I'll directly do it. Yeah, this is the last run. After that you can do. You want to leave? Leave.
I want to.
You can go. It will take maybe 15-20 minutes more.